Welcome to Pharmacology of the Synapse. This week we will examine the endocannabinoid system. In part 1, we will go over the discovery and description of the endocannabinoid system. We'll go over the molecules and drugs that act on the synapse and the effects of cannabinoids on the organism. Cannabis sativa has been grown for at least 12,000 years, initially for fiber and grain. The first documented use of cannabis as a medicinal plant is in 2700 BC in China. In 1839, an Irish doctor, William O'Shaughnessy, introduced medicinal cannabis to Western medicine based on his observations from India. In 1925, international treaties restricted the trade and trafficking of cannabis along with opium. In 1936, the seminal film Reefer Madness was released, demonizing cannabis and connecting its use to psychosis and violence. This anti-cannabis viewpoint continued in the 1960s and 70s, with laws that make it impossible to carry out research on cannabis. In contrast to the previous timeline, the pharmacology of the cannabinoid system has only very recently been discovered. The cannabinoid receptor 1 was discovered in 1998, followed by discovery of endogenous cannabinoid neurotransmitters in 1992. The cannabinoid receptor 2 was discovered shortly thereafter, followed by pharmacological tools such as the first CB1 receptor antagonist. It was not until 2001 that endocannabinoid signaling at the synapse was elucidated. We will discuss the endocannabinoid system, which comprises the two cannabinoid receptors, CB1 and CB2, the endogenous ligands, or endocannabinoids, EA and 2-AG, and the synthetic and degradative enzymes that regulate endocannabinoid levels and actions. Both CB1 and CB2 are GPCRs that are coupled to the GIO system, which means that when they are activated, they initiate intracellular signaling cascades that lead to reduced neuronal activity. Cannabinoid receptors are among the most widely expressed receptors in the brain and are also expressed in the body. CB1 is the primary subtype on neurons. CB2 has lower expression in the brain, is also found on microglia, and is expressed in the immune system. The term endocannabinoid describes the endogenous neurotransmitters that act on cannabinoid receptors. There are two endocannabinoids, anandamide, or AEA, and 2-arachidonyl glycerol, 2-AG. Both of these neurotransmitters are derived from fatty acids. Anandamide is a partial agonist of CB1 and CB2 receptors. 2-AG is present at higher levels than anandamide and is a full agonist with greater potency and efficacy than anandamide. The synthetic and degradative enzymes that control the production and breakdown of anandamide and 2-AG are very important in the cannabinoid synapse. 2-AG is synthesized by DAG lipase, or diacylglycerol lipase, and is degraded by MAG lipase, or monoacyl glycerol lipase. Anandamide is synthesized by NAPE-PLD, N-acyl phosphatidylethanolamin phospholipase D, and degraded by FAAH, or fatty acid amide hydrolase. Why the extra emphasis on synthetic and degradative enzymes? It's due to the unique signaling characteristics of endocannabinoids, which we will discuss next. Cannabinoid actions at synapses are unique compared with classical neurotransmitters like dopamine. First of all, endocannabinoids are made on demand. That means they are synthesized immediately when required and not packaged into any vesicles for storage. No vesicles means no traditional mode of neurotransmitter release. Instead, these lipid neurotransmitters diffuse across the synaptic cleft. Second, signaling occurs in a retrograde manner. We will go over this more closely in the next few slides, but endocannabinoids are synthesized on the postsynaptic side and signal at the presynaptic side. Third, endocannabinoid signaling is terminated by degradation within the synapse. It is still unknown whether a transporter or reuptake mechanism exists in the synapse, but so far research efforts have not found one. Overall, the endocannabinoid system acts as a negative feedback mechanism at synapses. We'll go over its actions step by step in the next few slides. We'll take 2-AG as our example endocannabinoid. In this synapse, the presynaptic neuron activates the postsynaptic neuron, either by depolarization induced by activation of calcium channels or activation of metabotropic glutamate receptors. This causes DAG lipase to produce 2-AG from lipid diacylglycerol. 2-AG then diffuses from the postsynaptic neuron to the presynaptic neuron and acts on CB receptors on the synapse. CB1 is an inhibitory GPCR, so activation produces inhibition of presynaptic calcium influx through voltage-gated calcium channels and inhibits vesicle release through other mechanisms. Overall, this decreases activity of the presynaptic neuron. The lipid-based endocannabinoids cross the presynaptic terminal where they are immediately hydrolyzed by their degradation enzymes. For example, 2-AG, this enzyme is MAG lipase. This halts endocannabinoid signaling. Both excitatory and inhibitory synapses contain CB receptors. Activation of CB receptors results in inhibition of synaptic activity of the presynaptic neuron, regardless of whether the neuron is excitatory, such as a glutamate neuron, or inhibitory, such as a GABA neuron. Cannabinoid inhibition of presynaptic activity is described with the rather complex term depolarization-induced suppression of excitation for an excitatory neuron, and depolarization-induced suppression of inhibition for an inhibitory neuron. The depolarization part comes from the fact that the postsynaptic neuron needs to be depolarized by the presynaptic neuron for endocannabinoid synthesis and retrograde signaling to occur.